As the world population continues to increase, the demand for food and other plant-based products is expected to more than double by 2050. Increases in yields for major crops, however, have largely stagnated in most regions of the world, with changes in global climate conditions expected to curtail productivity even further. There's a growing consensus that meeting the goal of doubling crop yields over the coming decades will require the sustainable intensification of global agriculture. Such an effort will depend on collaborative research aimed at developing new crop varieties that not only yield greater crop quantities, but are also more resistant to abiotic and biotic stresses, including drought, high temperature, flooding, disease, and insect pests. Gregg Howe, an MSU College of Natural Science researcher who is internationally known for his work on plant resilience and how plants respond to insect attacks, headed to Japan earlier this year as a 2022-2023 Fulbright U.S. scholar to apply cutting-edge genetic technologies to the development of crop plants that will contribute to sustainable agriculture and food security. He is here with us today to talk about his experience and the research he initiated with his Japanese collaborators to conduct joint research focused on understanding and improving plant resilience traits, specifically those of the domesticated tomato, an important crop in many regions of the world. Hello, and welcome to the podcast.
Thank you for having me.
Can you talk a little bit about what the Fulbright U.S. Scholar Program is and how you are awarded one?
Yeah, you bet. The overarching goal of the larger Fulbright is to foster exchange and knowledge, research expertise, and cultural activities between professionals in the U.S. and citizens in host countries. The U.S. Scholar Program specifically is designed to send U.S. scholars, scientists, artists, other professionals to partner countries where they will teach and lecture, in my case, conduct research in my field of interest. So faculty members from universities who are interested would apply to the program. They would, give a pretty detailed description of what their proposed activities are. These proposals are evaluated at a number of different levels, and typically sometime in the spring, the awards are made, and it all happens about a year before the actual visit takes place. And as it turns out, the application deadline is in mid-September, I think. And so if there are faculty in the college or at MSU who are interested in this U.S. Scholar Program, there are resources at MSU, specifically through the International Studies Program, that can help faculty put together their applications. And of course, I would be happy to chat with anyone who has specific questions about my experience.
The Fulbright experience brought together your personal expertise in plant resilience with that of Professor Hiroshi Azura at the University of Tsukuba's Plant Innovation Research Center. What was it like working so closely with international collaborators, and what would you say are the most unique opportunities that come with living and researching abroad?
Yeah, that's a great question. I've had many collaborations over the years, including several groups in Japan. Most of these collaborations tend to involve, simple exchange of materials or reagents, maybe information, and they're typically conducted remotely. But I think, what is missing from those types of virtual collaborations is the opportunity to really get to know your colleagues on a personal level and to share, ideas and perspectives that might extend beyond the immediate goals of the collaboration. So in my case, being, integrated into Professor Azura's group at the University of Tsukuba, it gave me the opportunity to interact with lab members on a daily basis, to get to know them better, and just to share our common experiences and perspectives. And I think, these types of things really help build long-term relationships and hopefully enduring collaborations.
So your work in Japan was an evolution of cutting-edge genetic research done here at MSU on growth defense trade-offs, which are how plants allocate limited resources during their development and how they respond to stressors such as insect attacks. Specifically, you traveled there to examine these phenomena in the domesticated tomato. Could you talk about why this was an especially exciting direction of research to take and what your findings might mean for ongoing conversations on food security?
Yeah, you bet. For the past several years, my lab has been trying to understand the basic mechanisms that underlie something we call growth defense trade-offs. So the simple explanation is that plants that are actively involved in protecting themselves against various stresses tend to grow more slowly. And this can be a problem in agriculture because our plants, in agriculture face many stresses, biotic and abiotic stresses. And as a consequence, they grow more slowly and they often have reduced yield. So it turns out that the prevailing hypothesis underlying this growth defense trade-off is one of resource allocation. So the idea is that, the plant has only so many resources, and if they put them into defense, then it's going to take away what they need to grow. Now, our work in this area, up to now has been done with a plant called Arabidopsis. This is the sort of the white mouse of the plant world. It's a model system. It's not a crop. But we discovered a few years ago is that certain genetic modifications in Arabidopsis allow the plant to both grow and defend well at the same time, sort of disrupting this hypothesis about resource allocation. So we were really excited about that. And as a logical next step, we wanted to see if we could apply those types of genetic modifications to a crop plant. And we chose the domesticated tomato to do this work, in part because our lab has a lot of experience with tomato. Tomato is an important crop, in many regions throughout the world, as you mentioned. And then really importantly for the Fulbright experience is that Professor Azura's group in Scuba is really a world leader in applying and developing gene editing technology to tomato. So put all of this together and it made sense to try to strike up a collaboration.
What do you see as the next steps for this research now that you're back on campus?
Yeah, that's something I've been thinking about recently. During my time in Japan, we initiated experiments, these gene editing experiments in tomato, as I mentioned to you. We hypothesized that these modifications will allow the tomato to grow and defend well at the same time, but these things take time. So right now, the plants are being propagated in Japan, and when enough seed is available, we'll bring that back to Michigan State, and we'll start to look at these plants in more detail to really test, how well they grow in the face of stress. Our particular stress we're interested in is attack by insect pests. This is a big problem in agriculture. It seems to be getting worse with changing climate conditions. So this is the way we plan to go forward in the future and to continue to build this collaboration.
Looking back, what would you say was the highlight of your Fulbright experience?
Well, there were many highlights. It's hard to pick just one, but I would say, in addition to the collaborative science that I did, another big highlight for me in terms of the Fulbright experience was the opportunity to travel throughout Japan to visit plant scientists at various universities and institutions. And, this actually was part of my proposal, this idea of going out, meeting new colleagues, potentially striking up new collaborations. These types of activities are really at the heart of what Fulbright has been doing for the last, 70 or 75 years. One specific experience that really stands out to me was the chance to meet and talk with students and postdocs, so young scientists, to talk about their research projects, to share experiences about graduate school and about career development. And it turns out, due to the COVID pandemic, which really locked down Japan for several years, very tightly, most, if not all, of the students that I talked to had never interacted face-to-face with an international visitor. So this was really the first opportunity for them to share their work with somebody outside of their group. So I think it was very helpful for them, and for me, of course, it was a joy to learn about what they're doing and to really get inspired by these young, talented scientists who represent the future of plant science in Japan. So that was something that really stood out for me.
So, research aside, what was the most enjoyable, surprising, or memorable part of your visit that took place outside of the lab?
Yeah, well, that's also a great question. As rewarding as the research component was for me in the professional experience, I have to admit that some of my most enduring memories were things that I did outside of the laboratory. I was very fortunate that my wife and son were able to join me in Japan for most of the time. And living there for 5 months, we definitely felt more like residents than we did tourists, which is quite an interesting experience. The daily activities of shopping, driving on the left side of the road, attending cultural activities, the public transportation, all of this, on a daily basis, I think really opened our eyes to, alternative ways of doing things. And there are just, many things about culture in Japan that we love. My son, I have to say, he was enrolled in high school there, in a local high school. So for him, I think it was a real life-changing experience. All of his classes were in Japanese, and it was really amazing for him. And yeah, I almost forgot to mention the food. The food was absolutely amazing, and we definitely did a deep dive into Japanese cuisine. So these personal experiences were really memorable. We made many new friends, and actually we look forward to returning in the not too distant future.
What do you have coming up in the near future? Is there anything that you'd like to share with our audience?
Well, I would say that getting back to research, for me, a research career has been very gratifying, in part because it's always changing, and there's always the potential for new directions that were largely unexpected. And once you publish your findings on a particular project, then you move on to the next one. So we recently identified a new gene in Arabidopsis and also in tomato that we think is going to be important for controlling this balance between growth and defense. We're excited to see where this takes us, and so stay tuned in the future.
If people would like to stay up to date with you and your research, what is the best way for them to do that?
Sure, I'm affiliated with several units at MSU and in the College of Natural Science. All of the units have active communication programs, social media, newsletters, and the like, and they like to highlight activities and accomplishments from faculty, staff, and students. And so in my case, I'm affiliated with the Plant Research Laboratory, the Plant Resilience Institute, which is a new endeavor here at MSU, and the Department of Biochemistry and Molecular Biology. So check out the websites of these departments, and you can often find some of our latest advances there.
Well, it's been a pleasure talking with you, and I'd like to thank you again for joining me here today.
Thank you for having me, Jesse. It was a pleasure being here.
Yep. Thank you.